So I was standing here in 2009 when this institute was opened, and the audience was very different. There were a lot of, I think, 6 ministers and some neuroscientists, computer scientists, and so on. And the future Nobel laureate was also here, Anton Rüttinger. So let me start my story. There are 2 young fish, swimming in the blue Danube near Vienna. And there comes an old fish from the opposite direction and says, "Good morning, boys. How is the water?" And of course, young boys are very self-confident. They don't say anything, just pass by. And then a minute later, one turns to the other and said, "What the hell is water?" So this wisdom from David Foster Wallace applies to neuroscience evidently. We don't think enough about things that are taken for granted or what things are trivial. And neuroscience is in this direction now. And of course, the reason why it is because we didn't start studying the brain. Our interest in ourselves started with the psyche, the soul, and the mind, and then only later on, the brain. Now, this comes with a big problem and baggage, because we inherit a lot of problems from our predecessors, and also a framework how we should try to identify a problem and solve the problem. So this is a kind of thinking I refer to as outside-in thinking. Why? Because the idea there is that the brain is given to us by God, and the reason why we have that is to explore and appreciate the beauty of the world around it. And it helps a little bit against our anxieties, which is putting us against this big universe and the infinity of time. And we feel small, but then here are the answers. So what the brain is for, not the brain, but the soul is there, is to communicate with some extraordinary thing out there. So we have to use sensors to enjoy the beauty. We have to collect all the information from left or right and collect that information and do something with it. In other words, we have to decide how to act. So the paradigm that we inherited is something like this. We observe the world because the brain was made for observing the world. If we want to do something with that information, it comes to a spooky entity somewhere in the middle. You can call it decision-making or a box, and then that generates an output. Now, I will tell you why this is a hopeless endeavor. But the way how it came about, of course, is that we neuroscientists inherited a vocabulary from psychologists. Psychologists inherited a vocabulary from philosophers. These philosophers are everywhere, but mostly the British empiricist, who said what I just said, that our sensors are the things through which we understand the world. And this British empiricism was driven by, or was given a paradigm for them by Christian philosophies. And this is where the choices, of course, were important, because you have to make a good choice, or choose between good and bad. And if you make a good choice, then you go to heaven. If you know, then you go to somewhere else. So the idea is here, and the idea shows that indeed we have to understand all of these things what are out there, but then the problem is how you translate it into something that helps my survival. Because ultimately, the body is needed to survive together with the brain. So in order to survive, you have to act. In order to act, you have to take this information and put it into something that is called a black box, intervening variables. And there are many, many names here that are used independent who you are, which direction you approach the brain from, but they mean exactly the same thing. And then you generate an output. There is an alternative thinking, the one that I'm proposing, which is the goal of the brain is to maintain itself. The brain is an organ that has no goal in itself or no use in itself. It just organizes everything else that's in the body. So the primary thing that a brain can do with the help of the body is to generate an output. And if you live in a very simple system, such as in the middle of the ocean, there you can generate outputs without any sensors available for you, the probability of survival is much higher when you move than if you don't move. Sensors came secondary throughout evolution when it served to adjust the sensor. The movement will adjust the sensors to make a better use of the action. So the one sentence summary of my talk is that the utility of the brain is to generate an action and check and determine and monitor the consequences of the action, because that will help you to survive wherever you live. Now, these internally organized patterns, or looking at the brain from inside out rather than the other way around, means a lot of things. One of the most fundamental things is that what we usually were taught is that we learn specific things, and from the specific things we make abstractions and we generalize. The other way of thinking actually came from this culture about Gestalt, is that no, the brain always generalizes. There is nothing new I can show you that you will say doesn't exist, because we will anchor the unknown to the known, and then we'll say, this room is like. And I know that I'm in an auditorium and so on, even though there are many, many specifics that are different from the auditorium at NYU, but the basics are the same. If for some reason that corner becomes important for my survival or for my prosperity, then, and only then, I will put my sensors and look at it very carefully. So what happened over the years before us, before neuroscientists, that we inherited a lot of problems to be solved. And these problems are encoded in a 2-volume big book, which is the Encyclopedia or the Bible of Psychology. Who can guess what it is? Any psychologists are around? Anyway, so... William James. William James. Nobody else knows in this room. You are the winner. You will get a glass of wine. So in the end of the 19th century, he codified all these words that were inherited from British philosophers and said, this is chapter 1, chapter 2, chapter 3, and everything that is in this book and the chapters are describing us neuroscience. And there were terms that were made up by people who lived before we knew anything about the brain. And the list is long. I just put here a few things, and I put a connection with those ideas with the brain. And you can see this is the frontal part of the brain. I had to make it a little bigger than usual to put in, to make enough space that all these things fit in, because this part of the brain is the garbage can of the brain. This is called the prefrontal cortex. This is where, if you don't understand something about the brain, we say it is done, performed by the prefrontal cortex. And this is where our personalities and also something. And you can see there are a couple of funny names here: consciousness, right? Cognition, theory of mind, dynamic filtering, daydreaming. All of these things come together in this area. Now, there is a vocabulary. There are the neuroscientists who are supposed to go into the lab and solve these problems. That is, to find home to each of these words that were concocted by people who didn't know anything about the brain. So if you think about it for a second, that's a dead end. That's not the right framework. This is not the right strategy. It has a deeper problem, and the deeper problem is this. According to the empiricist view, the way how you have to perform your experiments is this. You present something to the brain, and you record something in the brain. And then you have a correlation, and then when you have a correlation, then you have an idea what that part of the brain can do. This is exactly what happened. People found home for memory. That's the hippocampus. People find home for emotions. That's the amygdala. People find home for decision making. That's the prefrontal cortex, and so on, and so on, and so on. And the list goes on. Now, the strategy, of course, is to find boxes. If you know something about graph or graph theories, you know these are the nodes. But in the brain, the good things are not in the nodes. They are in the interactions. And those are many, and they don't have a name, so we don't know how to name them. So the problem here is that if you do this kind of experiment, Hubel and Wiesel were given a Nobel Prize for this innovation. But the framework was given to them already. So what you do is present a rose or a stimulus, and then you record from some kind of activity, a single neuron, it could be an fMRI, Bolt signal, or it could be anything that is dynamically changing. And then you say, I found a fantastic relationship between moving the rose or the presence of the rose and the activity of this neuron. Therefore, this part of the brain has something to do with vision. It's almost true, but the next step is very difficult to see what is exactly vision. What does it mean I see? And then the problem here is that the experimenter who does this experiment has no grounding. He is in a privileged situation because he or she, or today it's called they, they're only individuals who see the world outside, knows what's happening in the outside, and what happens in the brain. In contrast, neurons that you are recording from have no clue whatsoever what's going out in the world. I hope you understand the big distinction and the problem is that neurons have no clue what's out there. All they receive is action potentials from all the other neurons, and they integrate it, do with it, and decide to make an action potential. But that's not perception, it is not vision, not anything. So how does the brain get informed about something more firm, or at least a second opinion? And the answer is there is nothing else available for the brain except knowledge about its own action, which is the output. So the other way to think about this is this. Namely, every single time the brain generates an action, it sends a return envelope to the sensor system, saying that if the sensors detect something in the world, the reason why there is a apparent change is because I'm the agent. I'm doing that change because I am moving my eyes. You can experience the difference. If you close one of the eyes, you leave the other one open, and you move your right eye at about 3 times per second, what you will see is that the world moves because you are doing it, but in a different way than just moving voluntarily your eyes. So this is called a corollary discharge, as a information that is available now in the visual area neurons together with the input that comes from a retina. So every neuron, everywhere in the brain, receives at least 2 types of information. One is about the ongoing or outgoing action. It could be a real action or just the thought. The thought is an action also. And what comes in. And the comparison of the two will train the brain that, the consequences of my actions can lead to something good or something bad. Okay, so this is a summary here, but the trivial demonstration of what I was saying is this. The only way to know that a stick in the water is not broken is not to have 500 cameras around and making photographs and many, many sensors, but just to move it. The moment you move it, you realize that what reality is. And if you don't have the ability to manipulate the environment around you, then you don't get very far. So, as I said, the fundamental goal of the brain is to predict the consequences of its own actions. In a very simple brain, such as the one in the imaginative brain on the side, the organism does something and registers the consequences. And it works very, very well in a very simple environment at a very short time scale. If you are in the right niche to your organism, your survival is guaranteed. If you put the simple animal into another niche, it's completely lost. Now, with the help of evolution, we make this organ, the brain, more and more complicated, and we put new loops, many, many loops between the output and the input. But the goal, evolution doesn't have goals, but in retrospect, it looks like there was a goal. So the goal of this exercise here is to make the complications so good that now this same brain, or the same kind of brain, can predict its consequences of its own action in a much noisier environment and at a much longer time scale. That is, you can ask, what happens if you do this and that? This is almost cognition. But there is one step missing. Namely, the next important step is that this brain gets so smart that now you can turn off the inputs from the world, and the brain can really do a play, namely, what if I do this? What if I do that without actually acting out? And this is called thinking, but it's based on exactly the same idea, the same mechanism, and the same machinery. So this is a framework that is very abstract. Let's test it somehow. So one way of doing it is contrasting different things, different words, different terminology in one area of the brain called hippocampus, which is something that many people at ISTA do, and this is Joseph's favorite structure also, that we have been told that mostly experimentalist, that the hippocampus is a very important structure because it helps you navigate around the world. So navigation comes into two flavors. The first type of navigation that you can call is dead reckoning. That is, you just follow the directions, you count the number of steps, and then the vector change, you make a right turn, and you made 2 steps, and you can calculate with Pythagorean rules that I have to go back where I came from. So this is exactly the navigation that have been used for many, many years by sailors. Christopher Columbus got to the New World using exactly this kind of a navigation. They were very carefully detecting the distance traveled, the directions they went, and they could get back to the home base by extrapolating from the end. Now, once you explore the world, and every square inch is explored, then you can make a map. Once you have a map, the map is useful because it's no longer egocentric. It's not only me who knows how to navigate, but I can give that map to anybody who can read a map, and it's an abstraction. And then it is a superior form of navigation once you have that. And this kind of navigation was discovered, the credit goes to Vasco da Gama, when they started to sail from north to south rather than east to west. And then it's called the map type of navigation. So in order to build a map, you have to explore the world first. You have to know everything about everywhere. And then the advantage of the map, if you have it, then you can go from anywhere to anywhere else, even if you have never taken that route. This is graph theory. Now, extraordinary amount of experimental work went into this. People who were working with humans, they had a different idea about the same structure called the hippocampus. And they said, the hippocampus is about memory. And interestingly, there are 2 forms of memories. One memory is mine. It's called episodic memory or egocentric memory. This is what happened to me where and when. And these are my collections of experiences, which is me. Without my memories, I would be just a zombie. I wouldn't have a self. So this is a very important thing. But in order to get to the second level, to know about abstraction, I have to experience from one direction, another direction, a third direction, many directions, the same thing. For example, the first time we meet a dog when we are a little child, then it's an episode. And when we see another dog and another dog and another dog, and then it becomes just a abstract dog. And that's called semantic information. And in fact, this is the kind of strategic division also in psychology that people call episodic memory and a semantic memory. Now, what our insight was and has been is that it's impossible that this structure does something different in a rodent than in a human. But you can conceptualize things differently. You said, well, what is navigation and what is memory? Maybe memory is a disengaged navigation. That is when the brain no longer have to actually make steps, or the animal doesn't have to make steps. You just navigate into the past, which was behind me, and navigate into the future, which is in front of me. And we can call it memory because we are traveling back into the past. And that's exactly what mental travel is, episodic memory is. And we can also extrapolate into the future, and this is called planning. So all of a sudden, words like planning, navigation, memory, they are strange words to each other, but they come together when we look at the classification from the brain point of view. So the left side of the picture is very famous because Nobel Prize was given for the discovery of these cells. They are called place cells by John O'Keefe, which means that there are neurons in the hippocampus. They fire in a particular part of the environment whenever the animal happens to be here. So if I walk through this particular spot here million times, then million times I go through, there will be an activity increase of one particular neuron or perhaps a dozen of neurons or so. And then in the entorhinal cortex, which is an upstream structure from the hippocampus, they have a funny pattern. It looks like a grid. Now, for navigation, what you need, a couple of things you need. So when you emerge from a subway in Vienna, then you have a map in your hand. The first thing you have to do is to orient the map in the right direction. You have to know where is north. So there are cells that are called head direction cells. And this is exactly what they do. They orient yourself and they anchor them. Oh, this is the Stefan Kircher, you see that, and then the whole thing is in place. The second thing is, let's say, what is the grid in the system? Where are the streets and so on? And in your map, you have usually a square map. It said A22 and so on. And then the third thing is to locate yourself on the map. And there is one more thing that is not here you need is how far. Because you have to decide whether you take a cab or you walk there, so you have to navigate the distance. So if all of these are together, bingo, that's fine, everything works very well. So let me just show you a typical example of a place cell. What you hear, or you don't hear, but you can see these dots are the action potentials of one single neuron. The animal is running around, and then you can see that the one particular quadrant of the circle, this neuron fires, that's its private place field. This is where the neuron fires. And you can also see that some of this is the same neuron, the same different spikes, and then you can see here they are different colors. Now, the interesting thing is that, as Joseph already mentioned, hippocampus generate this reference signal called hippocampal theta rhythm. This is about 7 hertz to 9 hertz. And then the funny thing that was discovered by John O'Keefe, again the Nobel laureate, is that the spike on the theta cycle has phases, and the phase on which the spike occurs, there's either a theta cycle, in about 140 milliseconds, the neuron can fire here, 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 here. There is a systematic relationship where the neuron fires, because when the animal is approaching the spatial field, then it fires at the late phase, which is shown here as well as it's shown here. And then when it moves, and then you move here in the middle, then the phase changes, and so on. The take-home message is that the brain is better than just representing this big field, because you can take a photograph or a snapshot of about 100 milliseconds or so, and in every single 100 milliseconds, you just look at the relationship between one neuron and the activity of many other neurons, and this relationship will tell you whether you are here or here or here. The resolution, this way, with the help of this chopping oscillation, increases down to about 100 milliseconds. Okay, so as I said, wonderful things here. There is an interesting parallel between navigation, action, and abstraction, and which we think is a cognitive operation, such as memory. Now, I made a little connection here. The interesting thing was that for about 30 years, there was a lip service between these two fields, meaning that, oh, we discovered the place cells. Place cells, of course, are allocentric, independent of me. There's no time, nothing is involved. It's a static thing. But somehow you give time to it, and you will get episodic memory. But that would be a diagonal in my previous figure. So that's not the right connection. The other thing is that if it is true that you can disengage from the world, you have to have a demonstration for that. So how do you go from navigation to mental navigation? Now, the person I mentioned already twice, John O'Keefe, is an extraordinary scientist because extraordinary scientists are explicit. Explicit means I'm vulnerable. And I'm telling you the theory, and you can attack me. So the theory says that if you have a place cell in your hand, and somehow you are able to freeze the animal in space and time here, then there will be some neurons in the hippocampus that should fire forever as long as the animal is in here, in the same Cartesian coordinate system, because that's the prediction of the theory. So we tested that in an interesting way. So this is a memory paradigm where the goal of the animal is to go to the right, find water, remember that I made a right turn, and the next turn should be the opposite. Very simple task. Rats and rodents are very good at it, that when I learned something here, I have to move somewhere else. Ethologists call the win-shift strategy, because if I exhausted the food sources here, there is no point going back there. I have to go somewhere else. And this is a hippocampus-dependent task. So when you are recoding from these animals, the only trick we did is we trained the animal has to run in this holding area or the start area for about 15 seconds, always facing the same direction. So what we have done here is we have done everything in our power to keep the external world constant and information coming from the body constant. So everything is constant, and we are testing the theory which would suggest that as long as the animal is running in a wheel, you will find a place cell and it will fire forever. That's the prediction of the theory. And of course, I'm showing you because there was not a single neuron that did that. Instead, we found something more interesting, I would say. Now, many of my pictures are complicated, but I try to explain what they mean. So here, one line is one neuron. And this is neuron 1, neuron 2, neuron several hundred here. And the red color means that the neuron is very active. So this neuron is very active at the beginning, this neuron is active at the middle, and this neuron is active here, when we selected the trials in such a way that this collection of neurons occur and when the animal is making a left choice in the future. So these are left trials. When the animal goes to the left, this is the trajectory. That trajectory means that there is a order of neurons from 1, 2, 3, 4, 5, 1,000, and so on. That the trajectory goes in one direction. Now, when the animal decides to go into the opposite direction, you don't need a lot of statistics to see that this pattern and this pattern are fundamentally different. There is a new trajectory. So in this n-dimensional space, I don't have enough time to talk about the beauty of the anatomy here, but the trajectory can go in any direction, but the form of the trajectory depends mostly on the initial condition. And the initial condition, whether I came from the right or to the left. Now, the initial condition is here between the two, and the two trajectories are fundamentally different. And what it means that we can make an intelligent prediction 15 seconds before the animal turns right or left. You can call it a choice or decision making, with 90% accuracy if you have enough cells, including the errors that the animal makes. So think about this. That is at least 15 seconds earlier we can tell which direction, then which things the animal will decide. So this is a step forward to my claim that this is externalization and also disengagement of from the external world, because the same neurons that they look like place cells because they fire only a short period of time. In fact, if I give this data set to anybody who is working on a place cell, will tell me that they are standard place cells, except there is nothing in the world, nothing in the outside world that drives it. It's a self-driven, it's a self-organized change, an internally driven change in the brain that is moving things forward. Now, we can go one step further. actually several steps further, like in this situation, is that we will look at how the patterns, when the animal is not doing anything, relates or not doing exploration in the world, how does it relate to this, you can call it spontaneous or idling patterns of the brain. So here's another experiment that I'd like to explain. These are 13 neurons, and every tick is an action potential, and they are lined up in the direction of the animal's movement, and you can see that this is the first neuron, first place cells, this is the second place cell, and so on. There are 13 place cells here. They have a sequence, which is not a miracle. This comes out from the O'Keefe observation, but the funny thing that happens is that we look at the sequence of here at the long time scale, and we can see that within these theta cycles, there are similar sweeps of activity, but it's faster. So neuron 1 here, and neuron 2, neuron 3, and so on, then neuron 3, and neuron 4, neuron 5, and so on. So this activity replicates the long-term change. It is like a Bach's fugue. When you listen to the fugue, then you take a slice of 100 milliseconds or a short period of time, you can make a playful projection is what comes next, or especially in Greek canons. Greek canons, you can really predict how the long-term change will be predictable from the short-term changes. Now, even spookier, it turns out that before, this is during running, so this is cut out from here. But if you see that the same neurons that are active here, they're active also here and here in a very short time scale. And this is the pattern that the only thing you have to remember from my talk is sharp wave ripples. This is what Joseph emphasized, that this is a funny pattern because now all the neurons that are working one by one or relatively loosely with each other, all of a sudden come together in 100 millisecond time and they play the entire music. That is the entire trajectory. And you can see here that within this 100 millisecond time, this is called the ripple because it's about 150 hertz. And then the sequence of these neurons is pretty much the same as the neurons that unfold when the animal is moving. But the animal is not moving here. In fact, it's completely immobile. And that's the sequence. Now, at the end of the journey, even more interesting what happens, it goes backwards. So the same neurons are active again, but now in the reverse manner. Now, from this, you can make a sweeping conclusion. During the hippocampal theta cycle, the past, the present, and the future is replayed or played out in a continuous manner. There is no now, there is no past, there is no future. Everything is exactly the same. It's just one trajectory. Moreover, this trajectory is playing out before you start the journey. So this could be a preconscious or subconscious plan, if you want. Because this is the plan that you are trying to make. These are the sequences of neurons through which you have to go through. This is reverse kind of thinking. So the neurons don't fire because they are instructed at every single time that there is a constellation of the environment is changing. The hippocampus is not responding to the world outside. It already has an internally generated sequence. And the proof is here that before the animal actually did anything, the sequence is already there, pre-existing. At the end, and then you can call it planning. At the end, the sequence goes backward, and you can say, now the animal or the mechanism recapitulates what happened in the past. And you can call it memory. Actually, I don't know if you have time, but we can hear this. So there's a sharp wave. So you can see that this is what you have seen. It's a very nice sequence. And then the animal starts running. You can see from the rhythmic activity in a second. So here, and you can see also the mouse start running. So this is like Bach, right? You agree? And then at the end, we get to the end somewhere, and then here comes our sharp wave in the reverse manner. So that's interesting, because once you convince yourself that indeed the hippocampus is not a perceptive device that responds to the outside world, but it generates its own plans, and then it classifies the world according to the beliefs, if you want, or according to the evolutionary experience of the structure. Then you can ask this or say this question that calling the cells in the hippocampus play cells. And in fact, the other chapter that I didn't mention is that when the animal is running in a wheel, we can tell exactly how many centimeters or meters the animal was running, but we can also tell how many seconds elapsed in time if we compare it with the clock. So depending whether I'm interested in time, I'm interested in place, I can call it a place cell, time cell, but there are many other dimensions that the hippocampus can respond to. So in that sense, we can say it's irrelevant because it depends on how the experimenter sets up the design and how it interprets the findings. And if you confine things into a narrow possibility space, then you will always get your answer, what you are looking for. But if you look all the experiments that are done in different laboratories, then you can say the hippocampus can say, oh, this is time, space, or many, many, many other things. So an interesting problem here was that in order to identify what episodic memory is, you have to start with the definition and find elements of the definition, preferably in the same structure. So what are these? A mental travel happens with an event that is anchored to a place and to a particular time in your life. What happens to me where and when? This is the simplification. So now I took out time, I took out place from the equations, because I said these neurons are not place cells, these neurons are not time cells, they are just generic cells. Then how do we find episodic memory? And then from the brain point of view, you can say, maybe it's the other way around. The hippocampus doesn't do very much, except it generates enormous amount of possibilities. It can generate trajectory after trajectory after trajectory. And it can do it extremely well because the largest connectivity graph in the brain happens to be the hippocampus. So here, every neuron is connected to many, many other neurons. There are, in the red, there are 300,000 neurons, and they are connected to about 50 million possible connections. It's a very strongly connected graph. And then from any point to any other point, you can get into 100 milliseconds, which is across two synapses. So indeed, without any knowledge, the brain can generate enormous number of patterns. So the brain's fundamental occupation is to maintain this dynamic all the time. So if the answer is not what happened to me where and when, then you can say, well, what is the role of the hippocampus in all these complicated processes? And you can say, the hippocampus is a librarian. The hippocampus is a search engine. The role of the hippocampus is not to accumulate knowledge. The knowledge, the semantic information, is out there in the neocortex. The goal of the librarian is to point where the information is. So it's only a sequence connector to say, if you look at these books from the 19th century to today, you will understand history better than if you search and read randomly. And I skipped a lot of proof for that, but I'd like to show you one interesting experiment that shows this very effectively. This was an experiment done by others, my good friend Larry Squire in San Diego, who did the right thing, which is instead of asking questions from people who have missing their hippocampi because of disease or because it was taken out, then they were walking through the UCSD campus together with a group of people who were the controls. And then at the end of the walk, they were just asked simple questions. Then they asked, do you remember the fountain? Check. And then the amnesic patients were pretty good at many, many things. What they were lost immediately after the experience, and also much more after 1 month when they explained, tell me the order in which things happened. And then you can see from the red points here that they had no clue what happens first, which came after, because they had no sequence generator. They couldn't generate the sequences in the absence of the hippocampus. Now, with this knowledge, I hope at least you understood part of it, what I was saying. We can go to the next big question of the brain, is that how the brain knows. And what are the fundamental requirements and organizational principles that allow the brain to know? So one approach is that the brain is a randomly connected graph. There are so many neurons, and there are these rules that I don't have time about to talk. The inhibition and excitation is balanced nicely. There is, in order to make the brain do something, you have to put in noise. This is what the crutch of computer scientists are, or some computer scientists, because without the noise, no AI system works, and so on. And none of these models have any assigned role for sleep. Sleep is just an artifact or an unnecessary kind of thing. Now, the other kind of thinking is that the sequences are generated autonomously. Then the connections are strongly skewed. That's a big problem. And sleep is an essential part of this entire dynamic. And of course, the most important thing is that if you have such a brain that has so many patterns, then you can say that it's not what usually we think, that the world imprints information on my brain, and I have representation of what is out there, but it's the other way around, that my brain generates possibilities, and then I can unmask those possibilities and match through experience to something out there. So just to illustrate that the left side is not a completely old knowledge and useless thing to attack. One of the most famous people in the AI business said exactly that, right? That the brain is a tabula rasa. Now, another giant in our field is John Hopfield, who gave the complementary model, which is the Hopfield model, saying that we can implement memory mechanism very easily. We have to do a lot of things, but you need a large number of simple equivalent components, which is absolutely not the case in the brain. Everything in the brain is very skewed. There are neurons that fire 100 times more than, say, I'm not talking about different types of people. I'm talking about 100 Austrian 40-year-old males. They are 100 times different. They are not, but the equivalent in the brain, they are very different. Connectivity strengths are 2-3 orders magnitude different, and so on. Everything is skewed in the brain. The good thing about this is that it gives us a nice insight into one of the fundamental laws of psychophysics or neuroscience, which is called Weber-Fechner law. The Weber-Fechner law is a logarithmic law which tells you that what the brain sees is a difference based on the baseline. It's also always a proportional thing. If we switch off the light and somebody lights on a match, we will see that. Now, in order to have the same feeling in this room, you have to have a proportional change of the current light intensity. So larger has to have a proportionally larger change in order to perceive it. And it's not by chance that the brain thinks this way. And of course, it applies to memory. It applies to the number of steps we can make. It applies to many things that happens to us also in the dynamic, not only in the static sense. And the reason why we can do that is because in the brain, there is a logarithmic distribution of synaptic waves, firing rates, and so on, and connectivity. So if I look at the probability of my area, one of my areas in the brain connected to others, then I can say half of the wires are connected to a dozen or so other structures. It leaves the other half interesting because the other half is connected to a very large number of other structures weakly. Okay? So the same thing applies. At this moment, about 10% of your neurons are active. And they are good enough to comprehend some of the things I'm saying. If you would like to get to learn every single piece of it, you need to mobilize the remaining part. So what I'm trying to say is that there is a group of neurons, let's say 10%, that are responding at a very high rate, and they generate 50% of all the spikes in your brain in a given amount of time. but that's only 50%. The other 50% of the spikes are coming from a very, very, very large number of neurons who are just ticking along at a very slow rate. So that has consequences because there are fast-firing neurons and slow-firing neurons. And if I ask you to make a bet whether what happens to the fast and the slow-firing neurons tonight, or in another situation, 90% of the people I surveyed in the past, they said, oh, yes, many neurons are firing fast now, and then they get a rest in the afternoon, and other neurons will take over, and so on. And that's not the answer. The answer is that the fast-firing neurons will remain fast for the rest of your life. They are connected to each other better. They have information more effectively coming to them from the slow-firing neurons and so on. They form a rich club. And they are this minority that controls half of the knowledge of the brain. They are good in many, many situations. So just one example for that. This is all theory or discussion, but we have to anchor it to experiments. Now, the experiment goes something like this. The typical experiment that how we see that there are plastic changes in the brain, we put an animal in the home cage, in the box, we record from it while the animal is asleep and doing not so many interesting things. And then you put it in a task. And then you put the animal back into the home cage, and then you compare the two sleeps and say, what did change in the brain? And that change is presumably due to the learning that happened in the apparatus. Okay? So this is this is a graph here. What is shown here is the firing rate of different neurons. Every single XY coordinate here is a neuron. So this is neuron 1 to neuron 1, neuron 2, neuron 3, and so every neuron is cross-correlated. Every neuron firing rate is cross-correlated to each other. When neurons are firing faster, that it's not miraculous that they happen together in time with the action potentials of other fast-firing neurons. So that's not miraculous. What is miraculous is that the neurons that fire fast never or hardly ever want to fire together with the slow-firing neurons, as shown here in this very low, in fact, negative correlation. Now, to put the animal into the home cage, or the maze, and then in the home, sorry, I did show the home cage, but it's just before and after, and you make the difference, and you will find that only those neurons changed that are very low firing. The members of the rich club, the fast-firing neurons, didn't change at all. So all the plasticity comes from these very slow-firing neurons that are there and very difficult to find and very difficult to make statistics because they rarely say a thing. But when they say something, that's important. Now, another illustration of this log normal or skew distribution is the following. This sharp wave ripple pattern that I already showed you, this is the oscillatory pattern, can be short or it could be long. And we wondered if why is that the brain sometimes generates a short pattern, sometimes elaborates it even further. So what we found is that this long tail is probably very important because when we train the animals in a familiar or a new environment, then in the new environment, the sharp wave ripples are longer. Also, when we put the animal in an apparatus where the animal has to rely on memory or doesn't have to rely on memory, in the memory task, it is longer. Even more so when the animal has to perform one type of a task, sometimes it makes errors, then the sharp wave ripples before the animal makes a choice are shorter when the animal makes an error. So the longer the sharp wave, the better for you. That's the hypothesis. Now let's test it. There is a method. Some of your neuroscientists know this. What you can do is, in your retina, there are photosensitive neurons or photosensitive receptors. Now, these opsins can be expressed also in neurons. This is a big discovery in neuroscience. And then those neurons can be stimulated by light if you put a light source into the brain. So this is called optogenetics. So what we can do with the help of optogenetics is to detect, it's a closed loop system, where you detect the system online, the sharp wave ripple, and then you prolong it. You make it longer, because we think that longer is better. So that's the experiment. And then what we find is that those days when you prolonged the sharp waves and made them longer, the animals learned faster. So we we have done a decade ago to abolish or abort the sharp waves, and the animals got completely lost. So if you would allow me to take all your sharp waves out from tonight's sleep, you have about 4,000 sharp waves tonight. And every single one of them, I would cut it half. Then you remember exactly nothing tomorrow from any part of my talk. So this is a transfer mechanism that teaches the neocortex about sequences and what happened in the real world. So this is one thing, and I see my time is running out, but I'd like to show you several more things. But I skip this and go to this example. So I wanted to show you that there are neurons that don't fire a lot and so on, that we can make them fire and we can reveal and show indeed that they also have a knowledge about the world. They are just suppressed by other neurons. This is a different type of experiment where we ask the following. If the thesis is that there are pre-existing patterns in the brain that are giving you the opportunity to match them with experience out in the world, how are these patterns formed in the first place? And the good thinking is probably it happens during development. And so in this experiment, what you can do is take out from the womb, one by one, the fetus, and then go into the brain or into the ventricle with a pipette, inject something there, and then have electrical current to direct those molecules in the right direction to the hippocampus. In our sense, it's called electroporesis, but in fact, it's an electroshock to the animal. And then you put it back, and the animal grows up, and then you have a bunch of adults, and you can find those neurons that were born on your target day. So we have neurons that were born on day 13, day 14, day 15, and so on. So that's the experiment. And then you wonder whether neurons that were born together behave differently to each other than to other neurons. And you can guess from my preaching already, what happens is that neurons that are born on the same day, they like to fire together. They fire together also during the sharp wave ripples. Interesting thing that you are probably more interested is that those neurons, let's say we have 3 neurons here that have place fields. This is neuron 1, neuron 2, and the light blue is the third one. And they have some neighborhood relationship. Now you put the animal into the different environment, and you ask whether those neurons that are both together, whether they have the same relationship. And you will find that the blue neuron and the light blue neuron are firing at about the same place. And we know that these 2 neurons were born on the same day, and the red neuron was born on a different day. And then you do the statistics, and this is what you find. Now, all it comes down to is a conclusion so far, that the brain is a spaghetti. So there are so many patterns available, and these patterns are a realm of possibilities that the brain can do and gives you to match with experience. And you don't need a lot of plasticity here. In fact, Peter Jonas can tell you that there are inhibitory neurons in the brain, and with the help of a little inhibition, one trajectory, you are driving somewhere, and then all of a sudden there is a stoplight, and this trajectory can be diverted to another one. So you can concatenate, and the brain is like a Lego game that you can put together from pre-existing patterns, very complicated ones. So let's just summarize so far what happened, and you say, this is like, the brain is like a Chinese dictionary. My apologies to the Chinese here, but they are full with symbols that I don't understand. I have to anchor them to my known knowledge, which is English words or Hungarian words. And once I have them, I can match each of these symbols to that, and then all of a sudden the big vocabulary, or part of the vocabulary, becomes an intelligent piece of information. So everything is beautiful so far, I hope. There is a little problem. The problem is that I was preaching loud, and Joseph kindly said, many laboratories find what we found, and we are all obsessed about the hippocampus because it has this sharp wave pattern that is generating knowledge to us. But The sharp wave patterns are present in every single mammal, and even in an Australian dragon. And the problem with the dragon is that it doesn't have a big neocortex, but it has a beautiful sharp wave. So what the heck is doing, what the heck the sharp wave is doing there when there is nobody to read the content and nobody is to be taught, or not a lot. So I skip again a couple of slides, but I just go to this one, and which tells us that every single time there is a neuron here, somewhere in the hippocampus, it sends out a collateral and the pattern to the neocortex. And the neocortical readers are doing what I just told you they are doing. But the same neurons, they also have a collaterals. Many of them converge in a structure called the lateral septum, and the lateral septum goes to the hypothalamus. So we wonder if these many neurons fire during the sharp waves, it's almost 5 to 10% of the neurons fire together in a short period of time, they have to have a big impact on the hypothalamus. Now, it would be wonderful to measure many hormones and things that the hypothalamus releases. We don't have access to that, but we have a consequence of this, which is our blood sugar. So we can measure the blood sugar, and to make the long story shorter, we find that this is an occurrence of a sharp wave, and this is 30 animals. And when you have a sharp wave, then immediately, not immediately, the next 5 minutes after the sharp wave or sharp wave burst, the blood sugar goes down. So this is an interesting thing, and we have done a lot of experiments. We show that indeed this is coming from the hippocampus because we can make these sharp waves, and then we can artificially reduce the blood sugar levels. We can do another trick with opsins, is we block the lateral septum, and then we block the relationship between sharp wave activity and the glucose level, proving that this is a pathway through which these things occur. Now, the interesting thing here is twofold. One is that we have known from epidemiological studies that there is a strong relationship between sleep problems, obesity, and time to diabetes. And we provided a mechanism for this in an interesting way because the pattern that evolution initially invented for something useful for the body, such as regulate blood sugar and probably many other things, was accepted or transformed into something more interesting because the readers of the same information are now the neocortex. So the pattern was given to us hundreds of thousands of years ago. It didn't change, but the readership changed a lot, and that gives us something more complicated. So this is what we can call the dual use or sometimes triple use of things, that the sharp wave ripples work as a memory transfer. We have discussed how we can improve or deteriorate memories. Also, they change it in an interesting way. So to summarize, instead of looking the way the brain, how we have done it from the human egocentric point of view, that is, we are looking at from our cultural inheritance that the brain is there to observe the world, it's a little bit better to take an evolutionary point of view and look it from the body point of view. And if you'd like to have further reading, this is my recommendation. Thank you very much.